All right. Hello, everyone. Welcome to my special live stream. This is not my usually scheduled live stream on Saturdays. This, well, so this is for to discuss some science with Ed, who's been on the commoner on my channel and other channels, and he's basically been I guess, displaying a lot of personal incredulity, and it seemed like he doesn't understand the science. So instead of trying to going back and forth on in the comments, I suggested that he come on and we can have that discussion, um, you know, uh, directly, yeah, via voice, rather than trying to exchange it in the comments. So I'm going to bring him up shortly, but I'm going to try to make this. I want this to be a civil conversation. Uh, you know, I know in the comments it gets pretty toxic, but within here, I would like to have a civil conversation. But
Yeah, you can.
let me bring him on.
You bet. All right, I like it.
Hello, Ed.
Yeah, I'm here.
All
Can
right.
you hear me all right?
Yep, I can hear
Okay.
you. So, as a background, this discussion was supposed to be kind of discussing various different sciences, and you had given some conditions, some rules.
Correct.
So let me put up the rules. The first rule is there is no Fight Club. Oh, whoops, sorry, that's wrong. That's my other set of rules. Here are the real rules that Ed sent. So one is zero time wasted on talk about screen names or drama, like you said this about me, or religion. So we won't talk about religion, and we won't talk about Ed and his various alts that either say so and so blocked me or having names of serial killers. Second, 100% science only. Okay. Three, there will be no you are a liar without providing details as to what the lie is. So hopefully, um, when I get or we get accused of being liars, we'll have that proof. Um, there will be no running from a topic by claiming you have to provide sources, which seems to be kind of counter to rule number 3. You want the details to what the lie is, but you don't want to have to provide the sources. If you think I'm wrong, prove it. Again, that seems counter to number 4. And number 6, I'm going to make you talk about protein cellular structure, micro and macro evolution, abiogenesis chemistry, and that 2018 barcode study. I will have the paper printed off and we'll be reading directly from it. So I do. I don't think we're going to have time to discuss all these topics, but I definitely want to discuss the 2018 barcode study because I've spent a lot of time looking at it and have actually communicated with the authors, or at least one of the author, as I demonstrated in one of the videos. So that being said, let me hand it off to Ed. Ed, if you want to introduce yourself, whatever you want to say, you don't have to say anything that doxes you, but right now your camera is kind of like looking weird, but
Oh,
that's fine. Go ahead. You
yeah.
can introduce
I just
yourself.
have my thing laying flat. I'm
No,
on an
that's fine.
iPad. Um, First of all, I don't want to make this hostile either. The reason I'm on here is because what riled me up the most is the fact that every time I put something on there, post something that has science, nobody in your crew says anything about the science. It's always some kind of drama. It's always, he doesn't understand proteins, but never says why. I've never had anybody come back at me and say one single thing that was wrong about what I said. It's always changed the subject. And I don't really want to talk about the barcode study because that opens up drama. That's a he said, she said. I could read straight from the paper. You're going to say that's not what it says. And you're going to say you talked to the author, which I don't think you did. So how about we just get into proteins, which will lead to origin of life and abiogenesis.
So first of all, people not responding to you with science, can you imagine that it might be because of the way you post those comments, or is that not in your consideration?
No, my original comment is always just straight science. That's it.
Okay, people can go and look at the comments.
See, this is drama. Okay, I just want to get down to let's get right into the science. If you can disagree with me, you do it. If you can't, you can't. But we're not going to get into, you said this about this comment, or I said this about this comment. We're not going back scrolling through nonsense. Let's just get to it.
So I didn't
right
actually
now.
say that,
How
right?
about
I
that?
didn't actually like bring that up. I said people can go and look.
Yeah, but we're not going to
You
do
get
that
it?
now.
Okay.
We're
No,
not gonna do it now. Okay?
So when I said that, I didn't mean that. Okay, I'm going to share my screen and go through all those comments. I said
How about
people
let me
can
start?
go.
We'll start from there.
So
Okay.
let me just make one comment on the DNA barcode study. I'm not sure why that would be he said, she said, because we're actually discussing the research, the content. There's no he said, she said. And secondly, I actually like showed on the video my email conversation with the author, and I had actually asked you if you want to confirm it, just send an email to the author. But we won't talk about that, but I think we should talk about the research, because you had used this as really the only citation that you've ever
Okay.
provided to
Let
back up
me.
what you said.
How about we do this? I have the paper right here in front of me.
Okay.
Let me read the paragraph straight from the paper. Okay.
Well, tell me what
straight.
page. I can
Page
scroll to that
23.
page.
Page 23.
Page 23. Okay.
Okay. So it is. It starts with the last paragraph on 22.
Okay.
And then the first paragraph on 23.
Okay.
Okay. So it says that modern humans and it says they are basically between 100,000 and 200,000 years old.
So why don't you read the whole paragraph?
Well, it's a lot of stuff that it's unnecessary.
Well, it's relevant. It's relevant because
Okay,
that's
modern
what
humans are low average
yep.
animal species in the terms of APD. The
And
molecular
what
clock.
is APD?
I don't care.
Well, so you don't care about the data.
This is exactly what I didn't want to do. So I'm not going to participate if we're talking about this, because you want to argue about little stuff so that you can avoid not dealing with science. This is why I didn't want to come on here.
This is a science, dude. This is a science.
Look,
it's a paper.
Right here, it says, listen to this. Namely, that the extant population, which is the current populations, no matter what its current size or similarity to fossils of any age, has expanded from mitochondrial uniformity within the past 200,000 years. That means within the last 200,000 years, all species, 90% of them, had a mitochondria with 0 mutations. That means that they could not have been passed down from a different line.
So that's where you are misunderstanding the science, and that's why I keep saying that you don't know the science.
Okay, then why in the article does the author say we wanted to show there was evolution from a common ancestor, but the study did not? As much as we wanted to try, it didn't show it. In his own words. I don't know what to tell you.
Again, you quoted
I quoted an article that interviewed him.
Yes, but the part isn't the quote of him. The part that you are saying is him talking and I show that in the video where I demonstrated.
Look, I'm not getting into this.
I'm sorry.
I just read from the paper what it said.
I
don't know what to tell you.
Ask a question.
It just says all life, 90% of it is a uniform of mitochondria, and all life is roughly the same age and less than 200,000 years old. That's what that says.
Okay, go
I'm
ahead,
sorry.
Clark.
Are you claiming this paragraph we're looking at here is saying that all mitochondria of all animals
No,
is
no. All
200,000 years?
no. What it's saying is that within the past 200,000 years, 90% of all life that exists today has a mitochondrial uniformity, which means a point where there were 0 mutations, which is the starting point for that mitochondrial line within 200,000 years, which means that species started less than 200,000 years ago. And it even says,
Clark,
no matter, it doesn't matter what the fossil record says, it doesn't matter about any of that other stuff. It says
okay.
it started 200,000, less than 200,000 years ago.
So Blarg, I'm familiar with this paper, so let me just ask him. So Ed, do you know the methodology of how they determined this 100 to 200,000 years?
It's by
Or
mutation
is
rate.
that details?
It's mutation rate on a mitochondria. That's what it is.
So mutation rates of mitochondria. So how do they know what the starting point is?
Uniformity. That means they were able to
Uniformity with
track the
what?
rate. They looked at the species. They were able to track the rate of mutations, and they were able to, it's like rings on a tree. You're able to ring it back all the way to the beginning of the tree. It's the same thing with mutations.
So that's what you got out of reading this paper.
Okay, you tell me what I just wrote, what I just read about uniformity within 200,000 years and them saying that all life is basically the same age in less than 200,000 years. You tell me why that's wrong.
Sure.
Be specific.
I can do that.
Do
I
it.
can do that. By the way, do you
I'm
know
just
what?
aggravated because we're already on nonsense.
We're talking about science.
This is, I just read from the paper.
Yes, and we're discussing the paper.
Okay. Show me tell me why I'm wrong.
Okay.
Tell me why
So,
this paragraph is wrong.
So do you know what they barcoded when it says DNA
Don't
barcoding?
ask me
What did they
questions.
barcode?
Tell me.
Okay. All right. So I don't think you know it. So that's why I had
Don't
asked.
say, I don't think you know it. You explain it.
All right.
This is what I'm getting at. You constantly
so
avoid
I'll do that.
dealing with
I'll do
stuff.
that because, you know,
Being an aggravating asshole is not what we're here to do. We're here to be polite and civil.
I'm
You might want to just
trying
pull your head in.
because
You know you're not. No, you're not.
But it's
You're
frustrating.
you come in
because
like a
I
bull
know
at a
what's
that's
gate.
gonna
fine,
happen.
Blarg. I mean, I don't think it's worthwhile, you know, litigating that here. So Ed, I'm going to go and explain this to you. So I hope you can listen, and the other people who are watching, they can make their own decision.
Go ahead.
So what APD is average pair difference. And when it says, and when it talks about mitochondria, they're talking about a specific mitochondria gene. I think some oxidase gene. And the reason why they picked that is because it's very common across multiple different organizations. It's very commonly preserved. And so they're saying that in general, there shouldn't be any impact to evolutionary selection, meaning that you aren't going to get more mutational changes because it confers some benefit. These are all just point mutations that are neutral. So that's why they are quote unquote barcoding that particular gene. And so when they say that, that the clonal stage, there's a 1% sequence divergence per million years, which is consistent with the evidence for clonal stage of human mitochondria. So when they say clonal stage, what they mean is that where all the members of that species had the same sequence in that mitochondria gene. So the way that they determine when divergence occurred, when, for example, when the human species diverged from the chimps or whatever, or diverged from whatever their common ancestor is, it's by looking at the APD. Because what they're saying, starting from that clonal population where all the members of that population would have the exactly same sequence for that mitochondria gene, starting from there, they're checking to see, how whether the changes occur so that 100 to 200,000 years ago, years from then, there are enough changes that if you look at the different members of the species, so if we looked at your mitochondria gene for that, my mitochondria gene, and then, and that's what the barcoding is. And let's say you get 100 samples. And within that 100 samples, there will be some differences across the members. So what they found was that it's 0.1%, and 0.1% is equivalent to 100,000 years, meaning that every 100,000 years approximately, there would be 1 point change, 1.1% APD in that gene. And that 0.1% is like 1 or 2 base pair change. So it's like, it's very, very small. So what it means is that it wasn't created 100 to 200,000 years ago. There was a clonal population at that time where everyone had the same mitochondrial sequence. And clonal population, meaning that it's not 2 individuals, it just it's a small population. That's all it means. And so what the paper is saying is that if you do this for other species, you'll see that majority of them have only 1 or 2 intraspecies difference in the base pairs. And when they compare the interspecies difference, so if we looked at the mitochondria sequence for humans and compare it to mitochondria sequence for ducks, it'll be more than 0.1% difference. It could be like 1% or higher, meaning that the divergence did not occur 100,000 years ago, but maybe millions of years ago. And within this paper, it actually talks about interspecies APD as well as the intraspecies APD. So that's what this paper is saying. And you are taking that and misunderstanding because you don't even understand the methodology of the paper.
You done?
Yep, go ahead.
Okay. Phys.org, May 28th, 2018, sweeping gene survey reveals new facets of evolution. This is Stoeckle.
So you're not going to respond to what I said.
This is Stoeckle talking, one of the authors, and it says the study's most startling result, perhaps, that 9 out of 10 species on Earth today, including humans, came into being 100 to 200,000 years ago.
Okay, are you done?
Doesn't mean they evolved. And it said the conclusion is very surprising, and I fought against it as hard as I could.
Okay.
They were trying to prove evolution from a common ancestor. That's what they wanted to show. The study did not show it. Case closed. Okay, so that's what we're talking about.
Didn't say that.
It doesn't matter. I'm done. I'll just say you're right. I don't care. Okay. Let's get the proteins. Okay. Let's get the proteins, and then that's going to show why this is wrong. Okay.
Okay. So basically, you're not going to respond to me talking about the science.
I'm sorry. I really wasn't listening. I was
okay.
reading
So
the
you
paper.
weren't listening.
I was reading the paper and following along. But what you were saying was, it's not quite right. But it doesn't matter, because let's get the proteins, and then that's going to figure this out.
So you're going to say that what I said wasn't quite right, but you won't say I'm not quite right about.
I just gave you something from the paper that said what I said, and I just gave you a direct quote from the author that said what I said. Now, I don't know how you can disagree with that.
So you're saying that this article, and I have it on the screen, is more relevant and more authoritative than the paper.
I'm saying what comes out of the author's mouth is more relevant than what you think. Yes.
Okay. So if that's the case, then if the author says that this paper is not about it's about evolution, then you should take that, right? You should take this note that the author added to the paper about how this study is grounded and strongly supports Darwinian evolution, including the understanding that all life has evolved from a common biological origin over several billions of years.
And see,
so you accept that, right?
Okay, that's where words matter. Okay, nobody disagrees with microevolution, which is Darwinian. That shows up in the paper. Now, when it says it's strongly rooted or founded, that is our belief. That means that we believe that stuff happens, but the paper shows Darwinian. That's how that's read. It doesn't say both are grounded in the paper. One is grounded in the paper. The other one is a belief. That's how that reads.
Wow, words do matter. So what does billion years mean?
Billions of years means that's their opinion.
I see.
It is.
So their
Read
opinion,
read it again. Read it again, please.
this study is grounded in and strongly supports
See,
Darwinian
right? Okay,
evolution.
stop right there. Supports. Study supports Darwinian. And then what's the next word?
Okay, when it says supports, it means it's consistent.
No,
including
supports means it shows.
So in a science paper, they're not going to say this proves Darwinian evolution.
I didn't say proves. I said the data supports, which shows data was in the study. What's the next couple words?
It doesn't say data supports. It doesn't say data supports. The study is grounded.
When it
the
says
study.
The study
Okay.
it's firmly grounded in the study, right? Then they say another phrase. What is that?
What other phrase? Including the understanding that all life has evolved from a common biological origin?
Including the understanding. That's a belief. I have an understanding of things, but they're not saying it's in the paper. Words matter.
Wow. Okay. All right. I'm not going to argue too much more on this. I'll just bring up what you had mentioned about the paper, where it says here.
Let's move on. How about that?
You gave this. You
No,
claimed.
I wanted to start with the other stuff in the beginning. I didn't want to get with this first.
Well, then why did you claim that this is the author's quote?
I just read it out of the article.
And do you understand what quote marks are?
So you're saying that the author of the article made up a statement about what the author said, and he didn't really say it. Is that what you're saying? The guy published.
So the guy
published something that the author didn't mean. Is that what you're saying?
So it's not accurate, but it's, first of all, it's definitely not a quote of the author, which is what you
see. This is where it's quote, it's whatever. What he says and believes is right here in this article. It's in the interview. This is where you're trying to avoid stuff by arguing about nonsense like this. What is a quote? What isn't a quote? That doesn't matter. The article says this. And if he didn't sign off on it, the thing wouldn't be published. So
first of all, this is actually accurate. Then 9 out of 10 species on Earth today, including humans, came into being, meaning that the species came into being.
Exactly.
So prior to 200,000 years ago, there was no Homo sapiens. There was Homo heidelbergensis, which diverged into being a Homo sapiens.
Yeah,
so 100 to 2,000 years ago, that's what happened.
So I want you to use a little bit of logic here, okay? You've got millions. They did, I think it was 5 million samples, 100,000 species. So for us to believe that everything evolved from a different ancestor, then we have to believe that less than 100,000 years ago, 90% of life was something else and evolved 100,000 years ago altogether. Between 150 and 200,000 years ago. It was something else and evolved into what it is today, all at the same time.
No, it is definitely not saying that.
That's what it's saying.
It's saying that on average, species, the lifespan of a species is between 100,000 and 200,000 before speciation becoming another species. So it's not saying an event occurred 200,000 years ago that created every species that's alive today. It's saying that
it just says this in the paper.
No, it didn't.
It does.
No, it
didn't. Within 150 to 200,000 years ago, 90% of life came into being. It's clear.
No, it doesn't.
Let me finish.
Go ahead.
Okay. Please don't interrupt. Talk over the top of me. It said nothing like that. You're interpreting it to say that all of these species were just popped into existence at that time. All it's saying is that species have an average life of somewhere between 100,000 and 200,000 before becoming a new type of species. That seems entirely consistent with everything else we already know. And it's an interesting paper, that's for sure, that they found a different way to look at this and found the same information we already know.
Okay, one more time. The study's most startling result, perhaps, is that 9 out of 10 species on Earth today, including humans, came into being between 100 and 200,000 years ago. So it's saying that modern humans are less than 200,000 years old.
9
So
out of 10 isn't 100%.
That's what it says.
9 out of 10 isn't 100%, so it's not saying that about
It
modern
says
humans.
It says including humans.
It also makes assumptions about the mutation rate.
Well, you can argue with the authors then, not me.
But,
I'm just reading. What
and I did.
We
and
already know.
Well, you don't know anything.
Know that humans came into existence. Homo sapiens came into existence somewhere around 200,000 odd years ago. That's well established. And we know where they came into existence from.
Okay, I understand. This is why we
All
need
right.
to get the proteins, and it's just gonna make all this go away.
Okay.
You ready?
I
So I'll
hope
just
are you
moving
ready?
on to the proteins.
You ready?
Yeah,
I'm
hold
happy.
on. Yeah,
Okay.
we can do that. So Blarg, let me just, because what you said is correct. And the reason why it's 100 to 200,000 years, that range, is because, like I said, it's 0.1% APD. That's one base pair change. So 1 or 2 base pair change, basically the error rate for kind of determining the time is not like within, 100 or even 1,000 years. We're talking about gross 100 to 2,000 years. So it doesn't say that 9 out of 10 species all evolved within this small time frame.
So
It just
came into
says
being. Came into being.
yes, around speciated around 100 to 200,000 years ago, over possibly long periods of time. The point is that at that point, and that's what the author says, what they speculate is that there could have been some disaster at that time that reduced the number of population so that a lot of the population became kind of clonal populations, where it's a small population where they all had same consistent number of, or they had same consistent mitochondrial gene that without any difference. But we will stop here on this.
Okay.
I think we've said enough. So why don't you go ahead to the next topic?
So now I won't be hostile. I promise. This one just aggravates me. So this, what I'm going to bring up is why I know there's been comments about me being blocked, right? So this is why I was blocked by Aaron Raw, Creation Myths, and Professor Dave for this very topic. Okay.
But not by me.
No, but I'm having problems posting. Things just don't show up.
So that's not me blocking
on multiple. That's fine. I don't understand why they're not. I'm not going to blame you for anything right now.
Well, so that's why I said that, because you have been blaming me.
I was
blocked.
I was blocked, and they told
Would
me.
you like me to explain why that
And
is?
so let me get into why. Okay.
Get on with it.
Okay.
We don't need your preamble.
So in order for life to start on its own, and for it to evolve, proteins have to be randomly created by some random process. Okay. Now you take a single small 100 amino acid protein, there are 20 to the power of 100 possible configurations. That's 13 followed by 129 zeros. Only one out of that configuration will work with groups of proteins to do a job.
Please demonstrate that claim. That last one, that it's only one possible arrangement of protein that will work. Gotcha. That there aren't billions of other versions of proteins that could be quite similar or vastly different, but will also do a job.
Okay. I'll give you some examples. In humans, sickle cell anemia, okay, it causes a deformed red blood cell, and there is a single amino acid substitution, one amino acid substitution. There is proposed a single amino acid substitution that is the difference between modern human intelligence and Neanderthals. Every type of mutation we see, and it's been the DNA sequenced, you'll see a single amino acid substitution. It's that precise. It's that precise.
No, you
Wow,
have
that
to.
is
And so
very
Oh my
it's
gosh.
and why it has to be precise is because that's what causes the protein to fold in a specific way to do its job. You can't have multiple amino acid substitutions and it fold correctly and be functional. I mean, this isn't.
Do you know that there are over 50 different known alleles of just the proteins that are used for vision, color vision in human eyes?
Yeah.
There are over
I'm
50 different versions of the same protein that we know about currently. But apparently, if there was a single change to any one of them, it would stop working. So therefore, it would imply that only 2% of people should be able to see anything, and everybody else should be blind. That's not how that works.
Yeah. So what you're talking about is a late stage trivial color gene. Or
Okay, let me then give you a different example.
Let me just get into it. There's no what I'm saying is not controversial. Okay.
It is.
I know that you don't get it, but you look it up on your computer while we're talking.
Okay, so let me give you another example of variances.
Let me just keep going. Let me keep going. Okay.
Wait, so you're gonna just keep going and not let us respond to what you had said?
Well, go ahead.
Okay. So another example of, proteins that are, vary quite a bit is the immunoglobulin gene. And the way the immunoglobulin works is that you actually have to have humongous number of variances in the coding for the receptor piece so that, there would be something there that's going to latch onto some, antigen that's coming from outside, like a bacteria or a virus. So that gene itself has to have humongous number of variations. So it's not just one, base pair change. It's just like, I mean, it's actually designed to have different variations.
Yes, it is designed to be that way. Yes.
Okay.
But you
by who?
You take heart muscle, you take lung tissue, you take all of the stuff that makes things function. They have to be precise. And the reason why everybody's DNA, where humans are less than 1% different across the globe, is because everything has to be running correctly. And if you get multiple mutations, the person dies or can't reproduce. That's why we're so close.
You went to a different topic.
I'm not getting to a different topic. I'm talking about proteins.
Yes. I'm talking about immunoglobulin proteins. You said only one base pair change can ruin a protein because it won't be able to fold.
What you're talking about is the production of antigens. That's designed.
No, not production of antigens, antibodies.
Antibodies to attack antigens. That's
yes. So antigens are something different.
Yes, but it's to adapt to an antigen. That's the way they're designed. That's not a mutation.
So it's not a mutation that you have
that's a design.
Different
it's
sequences.
Designed to change to adapt to things. That's a design.
Yeah, that's how it came about.
I'm talking about you take 4 proteins that are supposed to work together to build something. They all have to work together. They have to fold correctly. They have to know their job. Okay. Now you look at the number of configurations for just a 100 amino acid protein. It is infinite almost. Now you got to think about if you have 400. If you have one part.
Again, you completely fail to understand the way that proteins work. And my example of the Rospin proteins in vision exactly demonstrate that. That doesn't matter because they're secondary proteins. No, I'm sorry, but proteins folding, there will be some certain elements within a protein folding that are quite important, and a mutation in that particular amino acid will result in a significant problem. But the vast majority of the protein structure, it doesn't matter what amino acid is in most locations in most proteins.
Who
It
told you
only
that?
matters in a couple of key points.
Who told you that?
Who told you that it has to be exactly right every single time? You give one example of one change and then extrapolate that across all of them. The reason we know this is because the example I just gave you, there are 50 different versions of that protein and they all work. So clearly,
Which one are you talking about?
True.
What are you talking about exactly that has 50 different versions?
Rhodopsin?
Rhodopsin protein in human vision. And that's not even counting the versions that are for color vision. This is just for black and white. So clearly, we can make multiple changes to a protein and not have it fail to work.
Okay, so let's just, even if there's 50, there's probably one amino acid change in 50 of them.
No, come on.
It's
okay,
true.
by the way, so in the hemoglobin gene with the sickle cell anemia, so you're saying because one protein changed that it doesn't fold anymore?
It folds differently. That's what causes the deformation of the red blood cell, and that's why the virus can't attack it.
And not the red blood cell, the hemoglobin.
The hemoglobin. It's the red blood cell that it's the part that can't get infected by the virus that normally can. It's the folding, the deformity that protects it, and it's one amino acid substitution.
So you are right that an amino acid substitution can change the structure of a protein.
The fold,
but not the
fold is what the function is.
The
fold is the function.
The structure comes from folding of the gene of the protein. So the structure can change with just one amino acid change. Mm-hmm. However, there are many amino acid changes, like when it's like alanine and valine, where they're very close amino acids where you can have that substitution and not change the protein, not change the structure of the protein.
You've got a protein that's 400 amino acids long, okay? You got to tell me that you've got to get, even if you've got to get 99% of them correct, you still have to get 395 of them correct.
No, we aren't saying you have to get 99% of them correct. I'm sorry, but that's not correct, Ed. It just
okay.
isn't.
How many do you have to get correct?
Imagine it this way: proteins have to fold in particular places to end up in particular shapes. They don't have to fold everywhere. Imagine you've just got yourself a folding table where you've just got a hinge in one location and a long piece of timber that needs to fold over on itself to fit somewhere. What that piece of timber is made out of is not that important, and it can be sort of moved around. It's just the hinge that matters. So if you've got another amino acid, you put a different piece of timber in that other piece of wood that's got to fold over on itself. It makes no difference to its fold. It's just the hinge. So in the case of proteins, they need to have certain amino acids in certain locations, but the rest don't matter very much or at all.
That's not true.
That's why the vast majority of mutations don't do anything. So
that's not true.
Well, it is true.
No, it's not.
What's it? My understanding is that somewhere upwards of 90% of mutations have no effect whatsoever on the organism.
I'm saying that a single amino acid substitution most definitely always does.
Do you understand how often?
Sometimes that's beneficial. I'm not saying it doesn't happen. I'm not saying it's not beneficial. It happens. That's how
Do you
you get a
understand
wolf
how
turn into a
often-
dog.
Do you understand how often mutations occur in your cells?
This isn't about mutations. This is about the creation of the proteins.
Well,
Now
we were talking about mutations because
400, in order to build a cell, you have to have hundreds of proteins form in the same place at the same time that work together to build the structure of a living cell.
Now
Because
we're shifting
proteins
the
degrade. You can't spend millions of years waiting for this one to build and the other ones hang around.
because that's not how it goes, Ed.
It doesn't. So where did the mitochondria come from?
you're moving
How
the
does the
goalpost
DNA get
again
made
now.
when it requires
So,
DNA requires proteins to make it?
So you're saying that at some point there would have been nothing, and then all of a sudden there would have to be a mitochondria protein?
I'm
Is
saying
that
that
how you're saying that?
I'm saying there's the machinery in a cell.
Mm-hmm.
It's made out of proteins. It's run on proteins. These proteins cannot be made outside of a living cell. There's not a single example of a protein more than 4 or 5 peptides that have been made.
Wait, so what's your point?
My point is that it's impossible for the proteins to come first, to then create a living cell that is able to divide and then begin evolution.
So let me see if I understand you correctly. Let me just see if I understand you correctly. You're saying that in order for...
Let me say it this way, and then I think it'll make it easy for you to ask a question. The simplest living cell that we already know has 473 genes, hundreds of proteins, the DNA, ribosome, the phospholipid membrane has approximately 30 different proteins. So all of this is required for the cell to be able to begin evolution.
Nope, not even close.
Wait,
Yes, it
so
is.
Clark, let me see if I understand what Ed is saying correctly. So Ed, what you're saying is that in order for abiogenesis to be correct,
Yes.
you would have to have a point where there's no life, and then you have to be able to immediately create a small, simple cell right away in one bang.
The reason
That's what you're
why,
saying.
yes, the reason why is because all the proteins to build that cell had to be created at the same time in the same place because they degrade. You can't spend millions of years. You can't spend millions of years just sort of making them and bringing them around and hoping they come together out in the water or something. They have to work together to build something. There are 60. There's roughly 60 different proteins in a ribosome.
So you're saying things that are completely irrelevant to
No, it's not irrelevant. You have to be able to build. Abiogenesis has to get to proteins because proteins build a cell and run a cell.
So how did humans create the first ship or first boat?
It started with humans. What I'm saying is if you don't have humans, you can't build a ship.
How did they build a ship?
If you don't have humans, humans are proteins. If you don't have humans, you can't build a ship.
That's fine. In the very first human who created a boat, how did they do it?
What has this got to do with anything?
What they didn't do was that
they took a log and then they strapped 2 of them together with a rope.
That's right. Perfect. So that is correct. What they didn't do,
it took intelligent design.
Come on, it's called an analogy.
I know it's a bad analogy.
It's called an analogy.
It's a bad analogy.
So let me at least kind of talk about the point so that other people can understand what I'm getting at. So what the humans did not do at the beginning in order to create a ship or a boat was to create a diesel engine, create, smelting for the steel that's needed to create the hull. They didn't do all those complex things. They started with something simple that's viable at that time under those conditions. So let's take the human out and talk about abiogenesis. So in abiogenesis, you don't start from nothing and immediately go to some functional cell with hundreds of proteins and all that. It doesn't even start from a protein. Have you heard of the RNA world hypothesis? Tell me what that is.
Yeah, nobody takes that seriously anymore because RNA degrades at room temperature within a matter of a couple hours. You have to store it in a lab at minus 85 degrees Celsius.
That's if you want to store.
That's why the vaccine from Pfizer for COVID, they had to store it. It's a RNA vaccine. They had to store it in a freezer at minus 85.
Have you ever done research?
I've been in the lab, yes.
You have been in the lab, and you have actually run protocols?
Tell me, does RNA not degrade?
Yes, so that's why I'm asking. So
I'm telling you that RNA reacts with itself, and it degrades at room temperature within a matter of a couple hours.
Come on, just answer the question. Have
What's
you run
the question?
protocols? Have you run protocols in the lab?
What does that have to do with anything?
I'm just asking, so you don't want to answer. So I'll take that as a no.
I've never worked with RNA in a lab. No.
I'm not talking about RNA. I said protocols. I didn't say RNA protocols.
Well, tell me what are you getting at? Don't ask questions. Tell me.
Well, I'm asking you questions to see if you can even understand this. So what happens is that in a lab, you will use reagents, and you're right. Those reagents
that didn't exist in abiogenesis.
That's fine.
On an early Earth. That's the point of it.
That's fine. That's right. In early Earth, there were no labs. Thank you. I agree with you.
There were no enzymes. There were no labs. No scientists. Nobody that could put something in there to keep the RNA from degrading. Yes,
that's right.
That's why there was no RNA world.
Yep. No, that's not why there's no RNA. So in a lab, you use reagents that are really, really pure. And you, of course, have to store them in good conditions because you're running experiments that depend on specific, concentration and specific effectiveness of those reagents. So you are, of course, going to store that in cold.
What reagents are you talking about? What reagents?
Reagents are anything that you use.
What is it that's going to be involved in the protection of RNA that you're talking about?
I'm not talking about protection of RNA.
We're talking about RNA degrading. That's it.
I was trying to.
Go ahead.
Okay, I was just trying to explain to you why your analogy doesn't work about comparing it to a lab, because it isn't a lab condition.
It wasn't an analogy. It was not an analogy. It's a fact.
No, but you're saying that in order for what you're saying to be valid, is that you would have to require some kind of like 99% pure RNA.
No.
In order for you to have this problem.
No. You can have 100% pure RNA. You put it at room temperature, it will degrade in a couple of hours.
That's what I'm saying. You don't need 100% pure anything in the primordial Earth.
So you can take 100, you can take 70, you can take 60, you can take 10. It doesn't matter because it's going to degrade in a matter of a couple of hours. That's why an RNA world doesn't work. It cannot substitute for DNA because it cannot store information because it degrades.
So how does it degrade?
It reacts with itself.
Reacts it with itself? How? What are you talking about? RNA reacts with itself.
Look it up. Everybody knows RNA degrades within a couple of hours. It has to be stored in a freezer at minus 85 degrees Celsius, unless it has enzymes in it that they put in the lab to keep it from doing it. This is common knowledge.
Go ahead, Barak.
I was just going to say, I'm sorry, Ed, but you had a set of conditions that if somebody wanted to naysay something, you have to prove it. Don't just say it.
This is something. You all have a computer. You have a computer. Look it up.
It's not my job to do your work.
There's nothing I can do to give you proof. There's no papers published on this. This is like asking someone to prove that multiplication works. It's basic stuff.
There's actually proofs of that. There's a proof of 1 + 1 = 2. There are actually studies of these things.
Yes. Now show me a paper on that. Show me a peer-reviewed paper that shows that.
That 1 + 1 = 2?
Yeah. Show me a peer-reviewed paper.
Okay, so I'm going to do what you did. Look it up.
That's my point. There's no peer-reviewed papers that says RNA degrades in 2 hours. That's just common knowledge, and that's why they have to store it at minus 85 degrees in the lab.
If
So
it was
do you
true,
understand?
there are definitely papers that say that. That claim is just utter garbage. There
Seriously?
would have, yeah,
Seriously.
yeah. It's utter garbage. It's an important piece of information
See, this is
that
this
we
is
need to know.
okay. This is where you need to step up and tell me why it's wrong. Where did you get your information?
I'm sorry, but you make a claim that it's common knowledge, everybody knows it,
Yes.
but it can't prove it because nobody's ever looked at it. And then when I
don't
say
have
that's
to prove it.
garbage, I have to prove it. Sorry, Ed, but that's just one rule for thee, one rule for me.
Okay.
So you're going to show the paper where it says that? And there are definitely many papers on RNA degradation rates in various different conditions, including intracellular and extracellular. So you can't say that there's no paper. You just haven't found any paper that actually backs up what you're saying.
AAT BioQuest posted February 8th, 2021. How long is RNA stable at room temperature? Answer: RNA is unstable at room temperature as it gets degraded within a half an hour. RNA
And what is the condition? Is it the primordial Earth condition?
It's RNA. RNA.
So you do understand that
at room temperature degrades within a half an hour at room temperature.
Under the current modern hemispheric.
So in order for you to have an RNA world, in order for you to have an RNA world, you have to come up with some type of a scenario that defies the chemistry.
No, it doesn't defy chemistry. It actually is chemistry in the sense that in certain conditions, you don't get that type of degradation.
What conditions?
What if it's not an oxidizing atmosphere?
It has nothing to do
What
with
if it's under very high pressure?
It has nothing to do with that.
Really? The degradation rate that you gave is under current atmospheric pressure and current atmospheric condition at room temperature.
So now you're suggesting that the atmosphere was a different pressure back then?
No, it could be
It
underwater.
didn't have a lot of oxygen.
It has nothing to do with water.
It certainly didn't have a lot of oxygen floating around in it.
Yes. When you actually look at, you can research that. Now they've gone back and they've said early Earth was very similar to now.
Oh, come on.
You can reach that yourself.
Show me that paper.
Yeah.
Can I make a
Give me the link.
Q?
Okay.
I can
Go ahead,
look.
This is what I was worried about. It's just like, this is common stuff.
No, there are
So everything I have to do, I have to show you common things. I can't get anywhere.
Yeah, I'm trying to explain to you that there are.
So I just read you RNA degrading within 30 minutes.
Ed, can you just hold on a second? Ook is talking.
There are multiple papers.
RNA samples are generally stored frozen at minus 20 degrees Celsius or 80.
Ed, Ook is, maybe you can't hear because Ook, I think you're kind of coming in quiet. You should
I didn't hear
talk
him.
louder.
Go ahead.
Right. There are multiple studies. RNA is stable at room temperature, exposed to air for 28 days.
Okay.
So what you're toting is either bullshit or you've misinterpreted the findings.
I'm reading off of a scientific site.
And so,
What are you reading off of?
So am I.
What is it?
And 92 weeks, at room temperature, it will start, it's still there, but it's not usable.
I didn't say it didn't exist.
You said it couldn't last.
I'm saying it starts to listen, let me be very clear. RNA has to substitute as a DNA. DNA has to store information that can be used to build, to pass on, to create new things.
Ed, we're talking about RNA, not DNA.
Yes, RNA world, that's what RNA world is supposed to mean. It means it was an early form of DNA.
It's not an early form of DNA. It's
Well, what was it
RNA.
supposed to
RNA is
do?
different.
What was it supposed to do? What's the point of an RNA world? What did RNA do? Tell me.
Okay, first of all, RNA is not DNA, and even in the current modern
I didn't
cell,
say it was.
RNA.
I didn't say it
So then
was.
why did you bring up DNA?
Because in an RNA world, it is used as a way to get to DNA. It is an early way they claim that it stored information and was able to create proteins like DNA was.
Okay, so have you actually like looked into what the RNA world hypothesis is?
Of course I have.
Okay, so what you said wasn't right. In the beginning, for the RNA world hypothesis, what they're saying is that you get kind of spontaneous short polymerization, where you get short sequences of RNA. And because they're short, it's not degrading at the rate that you're talking about, because length of the RNA actually makes a difference in terms of the degradation rate. Anyway, so you get these short polymers, just random polymers. So there's no enzymes or anything or templates directing a specific sequence. So out of those random RNA sequences, they have shown in the lab, of course.
In the lab.
In the lab, yes, because that's how science works. And in the lab, what they've shown was that they can find that that some of these sequences actually have enzymatic function or enzymatic, like functions like ligase and replicase and all that. So it's not strong; it's weak, but you can form or get enzymatic function from relatively short RNA polymers. So that's what starts first, not protein enzymes, but ribozymes, enzymes made from RNA. So that's what the RNA world hypothesis is, that you eventually get a ribozyme that is self-replicating, autocatalysis.
Okay, let me stop you there.
Okay.
Ribozyme. Self-replicating ribozymes do not exist. There was a paper that claimed it, but that was a lie. When you read the methods and materials, they took part A of one, they took part B of another. They made a highly reactive site, and they were both linear. And they both came together and formed a ribozyme. That's how they said they were self-replicating. Now,
Okay,
ribozyme a ribozyme has can be like 32 ribonucleotides. Every single one of those ribonucleotides has 16 different chiralities, and they all have to be
okay,
exactly
let's not get
the
into
same.
that yet. Let me just ask you about that paper. So that paper, when was it retracted because it was wrong?
It wasn't retracted. I'm telling you the materials and methods.
Wait, but you said it was like, there was some fraud or something, so it would have been retracted.
What I'm saying is the claim that it was self-replicating was in the title. When you go to what they did, it was not self-replicating. They took part A and part B. They created their own highly reactive site and connected part A and part B and claimed that it was self-replicating because they connected together. That's
okay,
what
so
happened.
it was a valid research paper.
No, because ribozymes fold. Functional
okay,
ribozymes
so it's not
are
a valid
folded.
science paper, so then it must have been retracted. When was it retracted?
I didn't say it wasn't valid. What
okay,
they
so
did,
it is valid.
What they did in the lab does not replicate a real ribozyme in humans or anything else. Functional ribozymes to do anything fold. So they cannot they cannot line up and duplicate.
Oh my gosh.
Do you not understand that part?
No, so you're still kind of hung up on this thing where you think abiogenesis is like modern proteins and modern ribozymes, modern cells just popping out. That's not what abiogenesis says. There are steps, different steps from one to the other. So you don't immediately like get into this, 200 sequence peptide being required. You can start with short RNA polymers that we know occurs spontaneously.
Do you not understand that you can talk about all these little things in abiogenesis and it's irrelevant until you get to the point where you have to make the things that are in a living cell? Right?
Yes, so that's what the RNA world hypothesis says.
Talking about an RNA world and protocells and all that stuff means nothing because you have to get to proteins. Abiogenesis has never produced a single protein in 30 years. They can't figure out how to do it.
So you have this condition of how abiogenesis has to occur. So, and if it doesn't occur in the way that you want,
okay,
that is somehow
claiming
wrong.
it works. You're the one claiming it works.
Not in the method that you're saying.
I'm saying
not in the method you're saying. Yes, you're right. It doesn't work in the way you're describing it. So I agree.
Okay. So how does abiogenesis create a protein in your way?
So when you have the ribozymes, so it has enzymatic function, and eventually it may get encapsulated into kind of lipid cells, small, primitive cells. And what that does is it just concentrates or increases the concentration of these molecules into like a small.
What molecules?
Into the lipid into the small cell, lipid cell. Like basically, it's just like a sphere.
What's the ribosome doing?
Sphere.
What's it doing?
Autocatalysis.
Of what?
It's just replicating.
Replicating what?
Replicating itself.
Auto means itself.
I need specifics. If you're going to go down this road, tell me.
Ed, listen. Do you know what the word auto means?
This is what I'm trying to explain to you. You cannot self-replicate. You cannot self-replicate a ribozyme because it is folded. It cannot unfold, split off, and make a copy of itself, and then refold.
Weirdly, papers on autocatalysis say that you're wrong.
Prove it. Give me one. Give me one.
Well,
give me one.
No, you made the first claim. Where's your paper that says it can't be done because it's all folded? It's always the same. You make the claim that it can't happen, and then as soon as someone else does something, give me the paper. And you go, I don't need to give you a paper. Everybody knows that. No, everybody knows that autocatalysis is fine.
A ribozyme, functional ribozyme, is folded. Everybody knows this.
So any sequence will fold. Any of these peptide and amino acids, nucleic acid sequences, they fold. Even DNA will fold. A single cell DNA will fold.
Yeah, but to self-replicate, this folded thing would have to unfold, make a copy of itself, and then fold again.
So do you know that when DNA is replicated in a cell, it is folded compactly and tightly?
Yep, and there are proteins that fold and unfold it.
Okay, so there are ways, right? Because it's folded
inside a living cell, that happens. Yes.
Okay, so in a relatively short RNA polymers,
you're talking about a ribozyme.
Ribozyme, meaning any RNA sequence that has catalytic function, is a ribozyme. You are talking about a ribozyme. I think you might be getting confused with ribosomes. But ribosome doesn't have to be that long.
I know exactly what I'm talking about.
So how long? What is the minimum sequence?
There are 32 in the paper that they did. There were 32 nucleotides.
So the paper that you can't cite,
the one you're talking about.
How do you know the one I'm talking about if you can't cite it?
You were talking about it just like me. You brought it up. I read it.
I brought up what?
The paper.
You don't even know what paper it is, and you said you read it. Okay, so go ahead.
That's me telling you the materials and methods, and I'm telling you there were 32 nucleotides in that.
Okay, so tell me what the paper is. Cite it.
I don't. It's self-replicating ribozymes. That's what the paper was about. There's only one.
Yes, so cite it.
Why?
Because that was one of your conditions.
Jesus fucking Christ.
That you're going to back up what you say. That was your condition.
No, you're the one saying it can self-replicate.
Okay.
I'm saying a ribozyme is it's folded. Every nucleotide has 16 different possible chiralities.
In that particular paper you're talking about, right? Not in general.
No, in real life.
So all ribozymes?
In a living cell, there are proteins that, and these things are constantly changing too, and there are proteins that fix them.
So are all ribozymes 32 base pairs?
I said in the paper.
Yeah, that's what I said. So when I said in the paper, you said no, in real
The
life.
number of ribonucleotides is irrelevant right now.
Why
The
is it
point
always like
is that
irrelevant when you're backed into a corner?
every ribonucleotide has 16 different chiralities. So if they all have to be exactly the same, and they constantly change. So you can
So do
have
you
5.
understand? Let me see if you understand this. In the RNA world hypothesis, you're not creating different sequences where, there's all these different varying chiralities, at least not that much. So in what they say happens is that you have to start with where you get biotic material, biomolecules. So meaning sugars, amino peptides, etc. Those things where it's, you're of course going to have different chiralities, but there are definitely ways that those chiralities are those molecules with different chiralities can get concentrated. So you can have like a concentration of a certain chirality, a homochiral concentration.
That's only theories, or that's all have a hypothesis.
So no, there's no data for that
But I
theory
know,
you're saying. So they just made it up.
yes.
So you're saying that these papers that talk about it are just fiction.
No, I'm saying they did some stuff in the lab, and they were able to come up with some stuff, but they're not able to say this is how it could happen in nature. Yes.
What they're saying is that they don't know exactly how that happened billions of years ago. What they're proving in the paper is that there is a chemical pathway, different conditions that lead you to these, steps that are needed to go to the next step in abiogenesis.
Okay, let's skip to the steps where life is created. That's my point. All of this other stuff is just nonsense. It's just stuff in a lab. Now get to the point where 200 to 300 amino acid proteins are created in groups to build the stuff in a cell. Until you can do that, abiogenesis means nothing.
So the first protocells, like I said, you get these kind of encapsulated ribozymes, and all
That's
they are
not-
really doing,
That's not
all
life.
they're really doing is
That's not life.
replicating.
That's not life.
It's replicating. That's how you start. So you get all these things that are replicating. You get, sorry.
That's
Sorry,
just made up. That's made up. That's just a hypothesis.
Ed, please give us a definition for life.
Um,
What does life mean?
A living cell is capable of a metabolism, its replication, it's able to interact with an environment. Those are just 3 simple things.
And let's have a look at the thing that QC was just talking about. Autocatalytic sets within a
they lack a metabolism.
A lipid
Metabolism.
bilayer. They are clearly getting energy from somewhere.
No, they're not. Nope.
So by the way, Ed, saying no is not an argument.
Then describe the metabolism.
Yeah. So
Krebs cycle?
why don't you first listen to what Blarg has to say before saying no?
Okay, I apologize.
Okay, thank
Go
you.
ahead. Go
Thank
ahead.
you.
Yeah.
Yes.
Sorry. Clearly, there is energy going into that process in order for the chemical reactions to occur. They can't just form in one direction. So they would only go in one direction if there was no source of energy. The modern idea of metabolism is it's basically the fighter jet engine of the way in which the system works compared to a sailing boat just going where the wind takes it. They're very different systems, but it's a process that still allows for very, very low speed, very low energy density systems to have these chemical reactions occur in a way that it has the chemical reaction go in the opposite direction to the one you'd normally expect it to happen. And you see that in these autocatalytic sets anyway. Whenever you get some of these molecules together, they can form into more complex molecules and create more complexity, which should be thermodynamically proscribed, I suppose is the way you'd put it. But that's not what we're seeing. You see the cells. I found another paper that suggests RNA lengths of 30, 60, 100, and even 120 pairs long being formed in an RNA world scenario. So for a molecule which exists in pretty much all living organisms as it is. So these are things that that happen. You're trying to ask for a definition of life that has is what we see in modern life. So this is the fighter jet version versus the sailing boat version. These are very different worlds.
So the point of it is, though, I'm sorry, the point of it is that all of that is irrelevant. All we have to do is it's life today as we know it. You have to get to that. It doesn't matter if you can get to a protocell, you have to get to the simplest form of life. That's 473 genes, hundreds of proteins, a functional phospholipid membrane that can regulate the internal environment, and a form of metabolism that creates some form of energy to power the cell.
So you can't build a ship unless you can immediately get to a diesel engine, a steel hull.
You're still going back to the analogy where you've got an intelligent design and someone
No, I'm
building
going
it.
to the analogy of where things progress step by step over long time.
Yeah, but in your analogy, then you would have to say that stuff floating down a river just happened to form into a boat and got more complex.
That's not what I'm saying. So I'm talking about how when you can't just immediately go from nothing to a fully functional modern cell.
So here's the problem you have, okay? Is that proteins cannot be formed outside of a living cell.
Wait, so we're talking about RNA. So
No,
this was even
has
before
nothing to do with
protein.
RNA. We're talking about life as we know
Yes,
it today.
life that started with RNA.
It doesn't matter what you think about RNA. You still even at 180 days, you still can't store enough energy. Information to pass on to replicate anything. In 180 days, you can't do anything.
What are you talking about? An autocatalytic set is one in which is inherently an evolving system.
Okay, how does it? But you still have to make proteins.
No, you don't have to make
start with
proteins.
So you never have to make proteins to
eventually
make
proteins.
Eventually.
Yeah, but that's not the first living cell.
Because eventually you also have to get to a DNA that works as a template. Because initially the RNA is a template. That's what autocatalysis is.
Yeah, but what's the RNA doing? What's it making?
Autocatalysis.
It doesn't matter. You can have 100,000 gallons of RNA in a lab. What's it going to do?
It's replicating. So you're
and
creating
it's replicating
units.
itself. What's it doing?
Yes.
But what's it doing?
It's
What's
replicating more and more complex versions of itself that are better at replicating itself.
And what are those versions going to do? What are they going to do?
More
doesn't matter if
replication
you've got more
of
complex.
itself.
Let me ask a very specific question. You're replicating your RNA. It's becoming more complex in your story. What is it going to do specifically?
So eventually it is going to kind of try to get to a point where it can create templates on DNA rather than RNA. Because like you said, DNA is more stable. We're not disputing that. What we're disputing is you saying that RNA cannot possibly start because it's just constantly degrading at room temperature.
That's a fact. That's a fact. That's a fact that it degrades. You even somebody read it. It's like
yes, it degrades, but it's about time.
Days. It had 198
Guess what?
days before it degrades.
So DNA is going to degrade. A cell is going to degrade. If you take a little piece of one of your cell and put it out, it's going to degrade. It's not going to stick around a long time.
Prokaryote.
Oh, really? So if you put a prokaryote Yeah,
If you put a
on just like
prokaryote in living conditions, it's not going to degrade.
so living condition as in what? Not like completely out in the air, right? In solution.
Not in something that's going to kill it.
So in some kind of solution then?
Whatever. Degrade is not the same thing as living, dying.
I'm talking about how you can, under different conditions, you'll have different degradations. Even a prokaryote cell is going to degrade under like non-solution conditions.
RNA degrades. It's not a viable DNA substitute. That's why the RNA world is not taken seriously. I don't know what to tell you. It's
I'm
just the way
sorry,
it
I
Ed,
is.
don't know
but
what
are
to.
you, Ed?
You
I'm
tell
sorry,
me. You
but are
show me
you
a
aware?
paper. Show me a paper where RNA sticks around for decades. Because
Ed,
you
are
have
It
to
you
think
doesn't
aware?
have
about.
to. It doesn't have to.
You have to think about inside your protocell. This thing has to be constantly building on itself, getting more complex without and outpacing the degradation that it's doing. Somehow
Are
it's
you?
supposed to get more complex while it's degrading. You tell me how that's possible.
So because
We
within
just
that
discussed it.
lipid sphere, it doesn't degrade at the rate that you're saying.
It
It's
degrades
not
with
room.
it reacts with itself.
Oh my God.
You didn't know that?
Go ahead, Blarg.
So you seem to think that an autocatalytic cell, an autocatalytic enzyme, is something that will break itself down. Autocatalytic means exactly the opposite. It's something that creates
An enzyme's a
new
protein.
copies of itself. That's what autocatalytic means. It creates new copies of itself. So it's clearly doing the thing you claim that can't be done. It's creating copies of itself, replicating copies of itself. If the old version breaks down after that, doesn't matter. It's got new copies of itself. It's got potentially better copies that are more efficient and more effective at doing the job that they were originally doing. The thing I did want to ask you, though, is are you aware that there are organisms today that do not have DNA in them, that only live with RNA?
What is that?
Bacteria. There's a whole
Are you talking about
pile.
the parasitic ones?
No, bacteria. There's a whole heap of bacteria
They're
that
parasites.
are they're RNA bacteria. They don't have a genome made of DNA. They have a genome made of RNA. They're living organisms, fully functioning living organisms that exist today.
Has everyone died?
I think Ed is looking it up.
Oh, okay.
Yeah, so it's not big, it's archaea. I don't know.
Well,
But here's the
there we
thing. Here's the thing is you're talking about RNA inside of a living organism. No one disagrees that RNA doesn't degrade inside a living organism. There are enzymes. So your point about it being RNA is irrelevant because you're already in a living cell. Your protocell doesn't have enzymes to protect the RNA like this archaea.
Autocatalytic set is
Do you not understand that RNA is degrading as it's being replicated? It's this auto thing, and it only it's got at the most 90%. It's able to copy at its most 90%, and it's degrading at the same time. You can't create complexity with those numbers.
Okay, so what is the ion solution where RNA is going to degrade within hours?
It's just,
or is it 100%
if you
pure
put it
H2O?
if you put it in any solution that doesn't have an enzyme to protect it, it's going to start degrading.
Okay, so you're saying in an ionic solution that it's going to degrade at the same rate as in a pure H2O?
Well, how much salt are you going to put in it?
I don't know. I'm just asking. So because you're making it seem like RNA is going to degrade regardless of the conditions.
So you're saying that inside the protocell was a highly was high salt concentration?
So what? No, I'm not talking about salt. I'm just saying different conditions. So inside that lipid shell, it's not going to be the same condition as outside. That's part of the reason why you have those lipid spheres, because it does create more, I guess, conducive conditions for cellular processes, including the RNA autocatalysis.
Do you not understand that the lipid is just a ball of fat, and it has
Yes,
no
and inside is,
proteins. It has no proteins for anything to come in or out.
however, it
It's a tomb.
protects what's
It's a
inside.
tomb. It's a tomb.
Yes, it
That's
it
it.
provides a constant condition for whatever molecules are inside it.
Yes.
Um, it,
And what are these things doing inside?
it's also
Self-replicating.
it's also not
Self-replicating, a degrading thing.
it's also not completely sealed from the outside. So a phospholipid bilayer,
We're not talking about that. We're talking about just the fat layer in the beginning.
they
Can't jump to
phospholipid
phospholipids.
phospholipids are a
Wait, hold on,
are
Blarg. Hold on. So Ed, you're saying you can just have lipids form spheres?
No, I'm saying that you're jumping from lipids to phospholipids.
Okay, so you're saying that in order to form spheres like my cells, you don't need phospholipids?
I'm saying you are jumping to phospholipids instead of just
It's not a
lipids.
jump. It's not a jump. You can't form
So
a
you're
lipid.
saying you're saying it wasn't simple lipids that formed this thing. It was phospholipids.
So you know
phospholipids
that
are self phospholipids are a don't have to be formed through the action of life. It's one of the molecules that we that abiogenesis researchers have already demonstrate form easily
In the
when
lab.
the right conditions are around.
In the
Which
lab.
conditions are always in the lab. The conditions they use are ones that are as close as they can get to what they expect certain conditions are. Phospholipids form. Phospholipid bilayers self-assemble into micelles. These are things that are well understood already. So yes, phospholipids, not
Okay.
just regular lipids.
So I'm still waiting for the production of proteins. This is what this is all about.
no,
Where are they coming from?
We said it starts with RNA.
But I'm asking you, I'm telling you that all of this trivial stuff you're talking about, this story, means nothing unless you can get the proteins.
So basically,
you-
So
all the
you can
steps.
make up all the stories you want about lipids and proteins, and I mean my cells and all of that stuff means nothing unless you can show how we could get the proteins.
Okay, why do you need the proteins?
Because that's what a cell runs on. That's life.
Because
Because
proteins
it
are
builds
enzymes.
everything. It builds
Because
everything.
proteins are enzymes.
Yes,
Right, Ed?
they're enzymes. There's 60 of them
They
in a
have enzymatic
ribosome. There's
functions.
30 of they do more than that. They form structures.
So
There's 60 of them
they
in a
have
ribosome.
enzymatic function because of the structure that they form.
Enzymes
Okay.
are like 60. They're very small. Enzymes are small.
Oh,
Proteins are
come on.
large.
Really?
What?
Are you wait, so did you graduate in microbiology?
You didn't know an enzyme's a protein and an enzyme is small? Small number of amino acids?
How many, so you don't think you can have large protein structures? Is that what you're saying?
Jesus. I'm saying that proteins are large, enzymes are small. Enzymes are around 40 to 60 amino acids, and most proteins are above 80. They can go all the way
So,
up to 28,000 amino acids in a human. That's how big a protein can be.
okay.
So I don't even know what you're talking about.
So did you study microbiology or not?
Yes.
Okay. So despite that, you're saying these things.
What did I say wrong? Be
So
specific.
what is a? So you're
Come on.
saying that beyond a certain size, you can't get an enzyme.
I didn't say that.
What did you say?
I'm saying that proteins, not enzymes, proteins are required to build the structure of a cell.
What do you call a protein? What? Because you're using terms that makes me really doubt that you graduated in microbiology.
You don't know what a protein is?
Do you? So what? What's the
A
difference
protein
between
is a
protein?
string of amino acids. They can range anywhere from about 80 amino acids to 28,000 amino
So
acids.
what's the difference between protein and enzyme?
Enzymes are small proteins, and
So it is a
they
protein.
don't build anything. They catalyze. This is two different things. Two completely different things.
So you're talking about structural protein versus enzymes, right?
Yes.
And they're both proteins.
I didn't say they weren't. I said they were.
Maybe you should go and listen to this after this. But
Let me be very clear.
Quick question, Ed. Ed,
You've got to have a ribosome to make
Ed?
proteins. There are 60 different proteins in a ribosome.
Ed,
Yes.
enzyme versus protein. You're saying enzymes are small little snippets that are a few things long, and a protein is a big thing. Is that? Am I getting that correct?
They're both proteins.
They're both. Yes,
Enzymes
but one is
are
small.
just a different kind of protein.
But you just told me said they were small, like 40 base pairs or 40
Enzymes,
amino
yes.
acids.
Yes, enzymes are small number of amino acids. Yes.
So what is amylase?
How many amino acids? Anything with an A's. Anything with an A's is an enzyme.
Is an enzyme. And hang on, how long is this thing?
I don't know what you're getting at.
It is a gargantuan molecule. How big is it?
Like 500 to 1,000 amino acids.
Yeah, it's a number quite a lot bigger than 40. And an enzyme. Let's just read the Wikipedia page. Enzymes are proteins that act as biological catalysts.
That's what. Yes.
That's it.
They
There's
don't build
nothing
structures.
about size.
They do not build structures. That's what I
There's
said.
There's nothing about size.
It doesn't.
In the word
this
enzyme.
This is a general. I said enzymes do not build structures. We're talking about proteins that build the structures in a cell, not enzymes. Proteins that build structures.
So in the RNA world hypothesis, you don't need protein structures. Really,
you
all
do
you need
to build a cell.
A cell forms with phospholipids.
Name a possible cell that doesn't have proteins. Name a function it can do.
Just a micelle.
Okay,
just a micelle with bunch of ribozymes.
And what are these ribozymes doing?
Self-replication.
And so what? Ribozymes inside something is not life.
They replicate.
Yeah.
If they self-replicate and create
what?
Improved versions of themselves as they're doing that, then that meets all the criteria of life.
No, it doesn't.
Yeah, it does.
The whole cell must replicate itself. It has to have a metabolism with chemical reactions. It has to be able to.
Why does a cell need metabolism, Ed?
To power all of the functions in it. If a thing is just in there, if a thing is in there and it's just got some stuff in there replicating, then all it is just some stuff in there replicating. It's not alive.
All those functions that the cell needs is to replicate. Ultimately, even your really only function is to replicate.
Essentially,
I have a metabolism. I can interact with my environment. I can change my environment.
These little cells, protocells, do exactly all of that.
Really?
They just do it extremely poorly in comparison to an organism that has all of the cellular mechanisms needed for what we refer to as modern metabolism.
Okay.
Modern metabolism is the jet engine equivalent of a paper plane. That can a paper plane fly? Yes. Does it need a jet engine to do it? No. But if you're going to be trying to fight a war with a paper plane against jet aircraft, you're going to lose. Which is why all modern organisms end up with these more complicated systems, because it makes them very, very much better at doing the things they do, which is basically self-replication.
Okay.
So the simplest thing is a self-replication.
It's a nice story, but get to proteins. Get to the part where we have life as we know it today.
So even though
I
we
just-
told you that the way this starts with RNA, you
that
just
is
want to go
just-
to
that is a
you want to go to the
hypothesis.
jet engine. You want to get to the jet
Hypothesis.
engine right away, right?
No.
Ook, what were you gonna say?
Sorry, it's sort of getting a bit long, the discussion and everything. Ed, what is your point? That
My point is,
no, I haven't finished speaking.
you just
yet.
asked me.
What is your point? Is it that we as humans don't know the exact mechanism of how or why the first cell started on Earth? Is that your main
No.
problem?
Let me sum it up with something that Jack Szostak said. I saw it in a video.
No, I actually want to hear you,
This
not
is
anyone else.
this is my point.
Right.
Okay. And he says it, and it's creation. Okay. He said, and this is what I'm trying to get at. The first living cell capable of cell division, the simplest possible living cell capable of cell division, must have a genome, proteins, form of metabolism, and a functional phospholipid membrane to control the internal environment. And they all have to be created at the same time in the same place, because none of those parts can exist outside of a living cell.
The last part did Jack.
The cell cannot be built gradually. That's what
So
he
which
said.
part of that did?
Hang on,
So
Q.
he actually said that they all have to occur simultaneously.
Yes, he did, because
Okay, all right.
because none of the parts can exist or be created outside of a living cell.
Okay,
So
all right.
That's my point with the proteins.
That's your point. But
proteins do not proteins cannot be created outside of a living cell.
Yeah, we get that. Ed, you do know that the only intellectually honest answer anyone can give to how exactly did life first start on Earth is we don't know.
We
Do you
know.
agree with that?
No, we know because of biochemistry and from what I just told you,
So
that it could not start on its own. Because in order for it to start on its own, it had to violate every law of chemistry and cellular biology and biochemistry that we know.
Ed, do we know exactly what the conditions were?
It has nothing to do with conditions. Nothing to do with conditions.
Oh my God.
Are
you sure you study biochemistry? If you're going to say stuff like that.
Do you not know that in 30 years of abiogenesis, they have tried every condition they can think of, and they cannot produce anything more than a 5 amino acid peptide?
Ed, I appreciate that, but you're saying you're going against every sane and intellectually honest person on Earth.
That's not true.
And saying you know what conditions were back then, and what are you positing as started life on Earth? Aliens, a greater being, what? Because as I said, and I'll restate it very clearly, the only intellectually honest answer someone can give to how life first started on Earth is, I don't know, but it's going to be fun finding out.
I didn't say.
Over to you.
I didn't say we didn't know. I don't know. Well, I'm saying I know how it didn't happen. And Jack Szostak summed it up. the parts of life as we know it today could not form separately and build themselves gradually by some random process.
Okay, so
There
let's
is
say
no,
that that's
there is
true.
no, there's no scientific evidence
Okay,
it happened.
sure, that's
There's
fine.
no,
Okay,
there's
so
nothing that says it
so
can't.
that's fine. Okay, that's
think
fine.
it's true.
Okay, so yes, it doesn't form randomly. Okay, now what?
What?
So okay, let's say we accept your hypothesis or what your claim, and I agree with you. Life did not start with complete random chemicals. So what? Now what?
Here, I know that you guys are all atheists, right? I'm not religious. I don't care. I just know that every professor I ever had in school asked these questions. Every one of them just said, I don't know. We just have no clue. So it got me thinking. And so I've looked into it more and more and more. And the more I've read and the more I've listened to people pushing abiogenesis, and the more I've looked into it, the more I realize it's not possible. And Jack Szostak, I even ran across that quote from him, and he admitted everything had to be created at one time.
Okay, Ed, if you're so sure of your position, where can we read your submission to a scientific journal?
See, it doesn't work that way.
Yes, it does.
I'm pointing out the things that don't work.
No, if
DNA cannot form outside of a living cell because proteins are required
do you know
to
how
regulate, to make it.
I know that. I've listened.
Proteins is 473 genes for the simplest living cell.
Now, Ed.
So I'm pointing out things that do not
Wait,
happen.
Ed, can you let Ook ask you to or make a comment?
I'm trying to
and
tell
then
you
can respond.
why. Okay.
But yes, we've heard that though.
Ed, you do know how the scientific community come to a consensus.
Yes.
People present papers for peer review
to prove something.
And
not prove.
They're
to
looked
prove,
at.
To try to show a hypothesis.
To try to disprove. That is how you.
No.
Really? So
you won't find you won't find a single paper out there with a title that says we proved this is wrong, or we did this to show this didn't happen. Never.
There's definitely papers.
No.
I'm sorry, you're wrong. Factually wrong.
then show
now,
me. Show me one.
If you are so sure of Ed, if you are so sure of your position, why haven't you submitted your hypothesis for peer review? It's a simple question. Why are you on here arguing with a bunch of atheists and not giving lectures at universities or writing more scientific papers that back up your original hypothesis? Why are you here
because I don't have a hypothesis.
Well, your hypothesis is that there's some intelligent designer that did this.
Nope. My hypothesis.
Okay, then that's fine, because that is, if you have no hypothesis, that's exactly what abiogenesis is. It's the study of how things could have come about. So you questioning it is essentially abiogenesis, but you don't have any data to support what you said. So everyone,
I have no doubt.
Hey,
I have no doubt that the chemistry fails.
Okay, all right. That's
not a hypothesis.
Your point.
That's not a hypothesis. That's pointing out a fact. It fails.
Okay. Yeah, that's called personal incredulity. So let me just
show me where I'm wrong.
Yes, exactly. All right. So we're way past the 90 minutes. Most of my streams do go a little bit long, but I don't want it to go on much longer, especially because I don't think we're making any progress. I think we're constantly getting, well, that couldn't have happened. That couldn't have happened. So, obviously, Ed, if you could send me a link or post a link to that Jack Szostak quote, I'll be kind of interested to kind of find out what that context is. But I'm going to wrap it up here. I know this has been frustrating, but interesting talk. So I'll have to, say, Ed, this was definitely interesting. And I'll have to say that, it was relatively cordial discussion, and it completely belies the kind of tone and the kind of things that you post on everyone else's and my channel. So take that as you will.
Amen to
Um,
that, Q.
And I'm going to just end it here. So thank you, everyone. Thank you, everyone in the live stream as well. This was, I hope you enjoyed it. And I guess I will see you in the comments.